Hello and good afternoon, friends. Welcome to the CEC-UGC live lecture. Dear friends, as you know that we have started with a new series on molecular biology, and so far we have conducted two sessions. In previous two sessions, we tried to understand about the various perspectives, the various important points in the molecular biology such as DNA, RNA, central dogma. And dear friends, I would like to tell you all that today also we are going to talk on our series on molecular biology, but today in this session we are going to discuss about DNA replication. We are going to understand what basically DNA replication is, and for this, dear friends, we have with us in our studios Dr. Varunendra Singh Rawat. He is an assistant professor in Department of Zoology, Hindu College, University of Delhi. So dear friends, let's welcome our guest Dr. Varunendra Singh Rawat, and let's try to understand about DNA replication. So first of all, I would like to welcome our guest Dr. Varunendra Singh Rawat.
Thank you.
Dr. Rawat, welcome to the EduSet lecture.
Thanks a lot.
As I already said that we have done with the two lectures on molecular biology. We try to understand about DNA, RNA, and today we are going to discuss on DNA replication. So one new term, that is replication, has been added to the DNA.
Sure.
So kindly try to understand, kindly try to explain us so that we could understand what replication
Sure, I will
is.
try my best. So, good afternoon all the friends. Today I am going to talk about DNA replication. It is a process which is present in all the living organisms as well as viruses. This process is very much important because it is required for continuity of different types of cells in an organism, as well as continuity of organisms from one generation to next generation. And the fidelity of this reaction is also very important, because if this particular process doesn't carry out properly, then there might be mutations in the DNA which will lead to various kinds of problems in the different types of cells as well as organisms. The importance of this particular process lies in the fact that this process is basically copying the information coded in the DNA in the form of genetic code. So today I would be discussing various aspects of DNA replication. I will be starting with the various enzymes which are involved in DNA replication. As the name suggests, replication is basically making a copy of a particular original molecule. Though the name suggests that this process might be very simple, just making a new copy of an original molecule, but it involves a complex set of proteins which ensure that the reaction or the process is very much conserved, and it leads to a molecule which is similar to the parent molecule. So I would like to continue with my lecture, and first of all I would like to give the overview of the lecture. I will be briefly discussing the history of DNA as the genetic material, how it was discovered that DNA is the genetic material. Then I would be talking about the experiment of Messelson and Stahl, which basically pinpointed the mode of replication of DNA. Then I would be briefly talking about the composition and structure of DNA, and the major focus would be on the enzymes which are used in replication. And at the last I would be talking about the synthesis of leading and lagging strands. So starting with the brief history of discovery of DNA as the genetic material, as must have been told in the previous classes, that Frederick Griffith was the first scientist who basically pinpointed that there is some basic transforming principle working as a genetic material. Though he was not able to say which material it was, whether it was DNA, protein, carbohydrate or lipids, yet he was the first scientist who was able to say that certain material might be existing in the cell which is basically the genetic material. He performed a set of experiments for establishing his hypothesis. He basically took mice cells and a virus named as Diplococcus pneumonia. This virus is basically causing pneumonia in the organism which it is infecting. So he performed a set of experiments. In one set, he took a strain of this particular virus, which is known as R strain. This is a strain which is non-virulent, whereas there is another strain known as smooth strain, which is a virulent strain. This smooth strain is able to able to dodge the immune system of the organism because it has a covering of the carbohydrate on its surface, whereas the rough strain does not have any such covering. Whenever an organism is infected with a rough strain, the immune system of the organism basically is able to kill this particular strain of bacteria. So, Griffiths injected different sets of mice with rough strain virus, rough strain bacteria and smooth strain bacteria, and he found out that the rough strain bacteria were not killing the mouse, whereas the smooth strain bacteria were killing the mouse. He did another set of experiment in which he took smooth strain of bacteria, killed them with heat and then injected them into the mouse. He found out that the mouse was healthy. That means the heat killed bacteria was not able to cause any infection in the mouse. In another set of experiment, he took the rough strain of bacteria and heat killed smooth strain bacteria. And when he injected these two components together, he saw that the mouse died, and also he was able to isolate smooth strain bacteria from the blood of the mouse. This experiment helped him in establishing that heat killed smooth strain bacteria had a transforming principle which could somehow transform the characteristic of the rough strain bacteria, and that led to the formation of smooth strain bacteria. Though still he was not able to pinpoint which of the molecule in the cell was the transforming principle. This pinpointing of the genetic material was done by an experiment conducted by Avery, McClure and McCarty in the year 1944. Avery, McClure and McCarty took live strain of bacteria and separated different components of bacteria, namely DNA, proteins, lipids and carbohydrate. They took DNA and along with it they took rough strain bacteria and injected these two components into the mouse. They again did similar set of experiment in which DNA was replaced with protein and injected along with the rough strain bacteria into the mouse. Another set of experiment involved lipid of these smooth strain bacteria, and this was again injected with the rough strain bacteria and injected into the mouse. Again carbohydrate was taken and with the rough strain live bacteria it was injected into the mouse. At the end of the experiment it was found that only DNA injected with the live rough strain bacteria was able to kill mouse, and live smooth strain bacteria were isolated from the blood of the mouse. Thus, this experiment pointed that DNA is the genetic material which leads to transfer of genetic information from one generation to next generation or from one organism to another organism. However, the scientific community still had its doubt, but these doubts were cleared when Hershey and Chase performed an experiment in the year 1952 involving T2 bacteriophage. T2 bacteriophage is a virus which infects bacteria and brings about the lysis of bacteria. Now, a bacteriophage has DNA as the genetic material and it has a protein coat which is present in the tail. The bacteriophage can infect the bacteria and inject its DNA into the cell and bring about lysis of the cell. Now, Hershey and Chase did an experiment in which they were able to radioactively label the DNA of the bacteriophage as well as they were able to label the protein of the bacteriophage. DNA was labeled with the P32 isotope and protein which has the amino acids have sulfur in them. So, sulfur was labeled with S35 isotope. They did two sets of experiments. In one set of experiment, they took bacteriophage which had radioactively labeled protein, but the DNA was not radioactively labeled. These bacteriophages were then used to infect the bacteria, and later on it was seen whether the radioactivity was transferred into the bacteria or not, whether the phage particles which were being produced afterwards, whether they had radioactivity or not. After the infection, the bacteriophage was separated from the bacteria by centrifugation, and radioactivity was checked whether radioactivity is there in the cell or not. Similar experiment was performed in which T2 bacteriophage were used, wherein DNA was radioactively labeled but protein was not radioactively labeled. After this experiment, it was found that radioactivity was present in the cells, in the bacterial cells, which were infected by bacteria which had radioactively labeled DNA. This confirmed to the scientific world that indeed DNA is the genetic material and protein, carbohydrate, lipids do not play any role in transfer of genetic information, though they may be having roles to play later on in different metabolic processes. By the year 1952, the structure of DNA had been unraveled, and it was found that it was a double helical structure. Watson and Crick suggested that the DNA replication must be semi-conservative, but still there was no experimental proof. Therefore, scientific world was of different opinion. The scientific community had given different hypotheses about mode of DNA replication, one of which was dispersive mode of replication. The dispersive mode of replication is basically when the two parental strands replicate, they form two new DNA molecules which had portions of both parental strand as well as a newly formed strand. The second hypothesis, which suggested that DNA replication is conservative, basically suggested that the parental molecule stays as such during replication, whereas a similar type of new molecule is formed which has been prepared newly. It does not have any component of the parental DNA strands. The third model, as proposed by Watson and Crick also, is the semi-conservative model of replication. This suggests that the parental molecule codes for the complementary strands, and one molecule of DNA gives rise to two DNA molecules, each one of which is having one parental strand and a newly synthesized complementary strand. However, due to the lack of experimental evidence, all the hypotheses remained in circulation. But the experiment conducted by Michelson and Stahl in 1958 confirmed that DNA replication occurs semi-conservatively. Michelson and Stahl took E. coli bacteria and they grew them in a medium which contained nitrate, which had heavy nitrogen, that is 15N in the medium. Therefore, the bacteria which is growing in the cell, that will uptake nitrate, and all the components like protein and DNA which have nitrogen in them, they will be having a heavy form of nitrogen. So, they grew these bacteria in this particular medium having heavy nitrogen, and they took bacteria from this particular medium and transferred them to another medium which had a normal light nitrogen containing nitrate, that is 14N. They performed the culture of these particular bacteria in this light medium for 2 and more generations, and after that they isolated DNA. In the first culture medium, where heavy nitrogen was present in the form of nitrate, when they lysed the cell and isolated the DNA and subjected it to ultracentrifugation, it suggested that all the DNA is having nitrogen, which is heavy, like it is 15 nitrogen. Whereas in the cells growing in light medium, after the first round of division of these cells, when they were isolated and they were lysed and DNA was isolated from them, after centrifugation it was assessed that this particular DNA was medium in heaviness, and it had one heavy strand as well as one light strand. However, the next step had to be confirmed, and this was confirmed when the second set of cells were taken after 2 rounds of replication. After the second round of replication, when the cells were lysed and DNA was isolated from them and subjected to ultracentrifugation, it was seen that the DNA after ultracentrifugation exists as two bands, one of which is very light in intensity, that is all the DNA is having 14N nitrogen only, and one of the band is intermediate in intensity, that is it is having 15N nitrogen as well as 14N nitrogen. This experiment suggested that the DNA replication is semi-conservative, because in the bacteria growing in the light medium, after the first round of division, all the DNA molecules were having heavy strand as well as light strand. Whereas in the second round, after the second round of division, there were DNA molecules which were having both heavy strand and light strand, as well as having only light strands. So this experiment established that DNA replication is semi-conservative in nature. Now the question comes that why should DNA replicate? DNA has to replicate because it contains all the coded information in the form of genetic code, which has to be passed from one type of cell to its progeny, like the epithelial cells which keep on dividing continuously. The information present in the genome should be transferred from one generation of cell to another generation of cells. This is done through the process of mitosis. Secondly, the organism have to pass their information which is present in their DNA into the next generation, that is the progeny, through the process of meiosis. Both mitosis and meiosis involve replication of DNA, and these particular processes have to be of high order fidelity, so that all the information which is present in the cells is passed on as such, and there is no change in the information from one generation to another generation. Now before I move on to the process of replication, let us have a brief summary of the structure of DNA. The structure of DNA as proposed by Watson and Crick is made up of a polynucleotide backbone. The backbone is made up of sugar and phosphate groups, and the backbone is made up of sugar and phosphate groups. There are two anti-parallel strands. That means one of the strands run in 5' to 3' direction, and the complementary strand runs in the opposite direction. The nitrogenous bases project from the backbone of the two strands, and they make complementary base pairing. There are 4 different nucleotides which are involved in the DNA. There are purines, namely adenine and guanine, and pyrimidines, namely thymine and cytosine. According to the Chargaff's rule of base pairing, adenine always pairs with thymine with the help of 2 hydrogen bonds, and guanine always pairs with cytosine with the help of 3 hydrogen bonds. This is the basic structure of DNA. Now the substrate for the reaction of this particular process, that is DNA replication, is a peculiar arrangement of double-stranded and single-stranded DNA, that is a primer template junction and deoxynucleotide triphosphates. The primer template junction is a region of DNA which has single-stranded template region and a small stretch of nucleotides which is known as a primer, which presents a hydroxyl group at its 3' end. The 3' end is the one which will be elongated during the process of replication. This 3' OH group is presented by the deoxyribose sugar of the pre-existing nucleotide of the primer. Now the basic substrate which adds on to the primer to give rise to a strand of DNA is deoxynucleotide triphosphate. This deoxynucleotide triphosphate has a deoxyribose sugar which is having attached to it at 5' position 3 phosphoryl groups. The group which is directly attached to the 5' group is known as alpha phosphoryl group. The middle phosphate group is known as beta phosphate group, and the outermost group is known as gamma phosphate group. To this particular deoxyribose sugars are added for the given 4 nucleotides, namely adenine, guanine, cytosine and thymine. Now these are the substrate for the reaction. The template does not undergo any change during the reaction, it just codes for the information for the formation of the new strand. Now talking about the chemistry of the reaction, how do new nucleotides get added to the primer which is already existing at the primer template junction? This is brought about by a nucleophilic reaction, that is SN2 reaction, wherein the 3' OH group attacks the alpha phosphoryl group of the incoming nucleotide. This particular reaction leads to formation of a phosphodiester bond between the primer, pre-existing primer and the incoming DNTP. The leaving group in this particular reaction is pyrophosphate group, which has 2 phosphoryl group attached to it, namely beta and gamma group. The delta G for this reaction is quite low, it is about minus 3.5 kilocalorie per mole, which is quite low for the reaction to proceed forward. However, extra energy is generated by hydrolysis of the pyrophosphate by an enzyme known as pyrophosphatase. This generates another minus 3.5 kilocalorie per mole of energy. Thus, the total energy for this particular reaction comes to be about minus 7 kilocalorie per mole, and this is sufficient energy to drive the reaction in the forward direction, which makes this reaction particularly irreversible reaction. Now, I will be focusing more on the enzymes which are used in replication. There is a series of enzymes which are required for effective replication to take place. The first among them is DNA polymerase, which is the essential enzyme which will bring about replication of DNA, and all the other enzymes help DNA polymerase in carrying out this particular reaction. The other enzymes are namely helicase, sliding clamp, ligase, topoisomerase and primase. DNA polymerase was discovered by Arthur Kornberg in the year 1957. He discovered this particular enzyme in E. coli and he used this enzyme for in vitro synthesis of DNA. However, this enzyme was not the main enzyme which is used in genomic DNA replication. This enzyme was DNA1, Pol1, which is used in repair mechanism in E. coli. However, this discovery led to the foundation, it was basically the foundation which led to discovery of various other types of DNA polymerase enzyme. For his discovery and contribution to this medical world, Arthur Kornberg was awarded Nobel Prize in Physiology and Medicine in the year 1959. After this particular discovery, a battery of enzymes which help in replication of DNA have been discovered in both prokaryotes as well as eukaryotes. In prokaryotes, namely 5 enzymes have been discovered which come under different families of polymerases. These enzymes are namely Pol1, which was discovered by Arthur Kornberg, which comes under family A and functions in excision repair of the DNA and also in removal of RNA primers which are formed during DNA replication. Pol2 comes under, it is categorized under family B and it helps in DNA repair. The third enzyme, namely Pol3 enzyme, is a member of family C of DNA polymerases and is the main enzyme which is used in DNA replication. This enzyme along with other set of enzymes form DNA Pol3 holoenzyme which together bring about effective replication of DNA in E. coli. Pol4 and Pol5 come under family Y and they are involved in repair mechanism, whereas Pol5 is also involved in translesion synthesis. Now, there are about 15 eukaryotic DNA polymerases which have been discovered and only 3 of them are majorly responsible for carrying out effective DNA, the genomic DNA replication. All the other enzymes are used for other purposes. Herein I will be discussing only about 4 or 5 enzymes which are namely alpha polymerase, beta polymerase, gamma polymerase, delta polymerase, epsilon polymerase and telomerase. Alpha polymerase comes under family B, and this enzyme is the initiating enzyme of DNA replication. It also has a domain which helps in formation of RNA primer during the initiation of replication, whereas beta enzyme is involved in base excision repair during the replication. The gamma enzyme is basically not involved in genomic DNA replication, it is involved in mitochondrial DNA replication. The two other enzymes, namely delta and epsilon, are the enzymes which carry out the major chunk of replication of the genomic DNA in eukaryotes, and they also have many other characteristics like excision repair, mismatch repair, etc. A special enzyme known as telomerase, which comes under reverse transcriptase family, is involved in replication of the telomeric end of eukaryotic chromosome. This is very important enzymes, because if telomere lengths start to shorten, then it might lead to loss of genes, and it will lead to serious consequences for the concerned cell. So, telomerase helps in maintaining the length of telomeres of the eukaryotic chromosome. Now, what is the mechanism of DNA polymerase action? How does it go about replicating the DNA, which is a double stranded molecule? DNA polymerase has one active site which is responsible for addition of 4 different substrates. That is, only one active site is responsible for addition of adenine, guanine, cytosine and thymine. How does it carry out this particular process? This it does because the active site does not recognizes bases as such. It just recognizes the geometry of the correct complementary base formation between the incoming base and the template. If a correct base comes and gets attached to the primer complementary to the parental strand, then the reaction carries further on. However, if there is a mismatch, then there will be excision repair system. Whenever a correct base comes to the active site, it fits into the active site such that the primer, which is having its OH group free, it attacks the alpha phosphoryl group of the incoming DNTP. This leads to a phosphodiester bond formation. However, if an incorrect nucleotide comes, that is, if it is not complementary to the nucleotide present in the template strand, then it is not going to fit properly into the active site, leading to improper positioning of this particular DNTP such that the OH group of the primer is not able to attack the alpha phosphoryl group of the incoming DNTP, and the reaction will not take place. Herein, the active site displays kinetic selectivity, which is enhancement of the activity whenever a correct substrate is present in the active site. However, if there is an incorrect substrate present, then the reactivity or the action of the enzyme is decreasing by about 1000 folds. Not only this, the DNA polymerase faces another problem, that is presence of similar molecules, namely ribonucleotides, which are used in the formation of RNA. Now, these molecules are similar in structure to DNTPs, deoxyribonucleotide triphosphate, except that the ribose sugar in RNTPs have an extra OH group. Now, the DNTP can fit properly into the active site and reaction can take place. However, because of presence of extra OH group in RNTP, there is steric hindrance of fitting of RNTP into the active site, because there are certain discriminator amino acids present in the active site of the DNA polymerase. When this happens, the RNTP which has come to the active site, the phosphate group is not properly placed for the reaction between the OH group of the primer and RNTP to take place. Thus, there is no reaction taking place, even though RNTPs are present about tenfold higher concentration compared to your DNTP. So, this is how RNTPs are not incorporated into the DNA strands. Now, coming to the three-dimensional structure of DNA polymerase, which has been ascertained using molecular data, it has been found that there are three domains present in DNA polymerase, which resemble a partially closed right hand of human beings. These three domains are namely termed as finger domains, thumb domains, and palm domain. Palm domain is the main domain which is responsible for catalysis of this particular reaction, whereas finger domain also plays a partial role in catalysis, and thumb domain, though it does not play any role in catalysis, it is important for this reaction to take place. We will see how catalysis takes place in the active site of the palm domain. The palm domain has certain special components like amino acid and specific metal ions which help in catalysis of this particular reaction. The active site has two metal ions which are divalent, and these metal ions play a very important role in catalysis. One of the metal ions displaces the hydrogen atom from the OH groups, which leads to formation of O minus. Now, this O minus makes a nucleophilic attack into the alpha phosphoryl group of the incoming DNTP. The second metal ion, it coordinates and stabilizes the other two phosphoryl groups, namely beta and gamma phosphoryl group. The active site is present in the palm domain in the crevice between the fingers and the thumb domain. Apart from using metal ions for catalysis, the amino acid residues of the palm domain make several hydrogen bond interactions with the minor groove of the newly forming DNA. This helps in stabilizing the primer template junction in the active site, and it also helps in fidelity of the reaction, which we will see later on. Now, finger domain also has a role to play in catalysis. Finger domain remains open when there is no DNTP coming to the active site, but once a DNTP comes to the active site and it makes correct base pairing with the complementary base of the parental DNA strand, then finger domain has certain amino acids which make contacts with the bases and the phosphate group of the incoming DNTPs. This helps in proper aligning of the incoming DNTP so that the 3' OH group of the primer can attack the phosphate group of the incoming DNTP. When a correct nucleotide comes in position, then residues like tyrosine make contact with the base of the incoming DNTP, while in this particular figure, lysine and arginine are making contact with the phosphate residues of the incoming DNTP. This leads to folding of the finger domain by an angle of 40 degree, which brings DNTP much closer to the 3' OH group of the primer, which effectively later on helps in catalysis, that is formation of a phosphodiester bond. The finger domain also takes part. It is also responsible for the fidelity of the reaction. It ensures that a correct base is incorporated against the template each time the primer template junction comes to the active site. Only one base is projected into the active site at one time, and this is because of the bend in the template of the DNA strand in the finger domain. This ensures that only one base is projected and the correct base is projected. Otherwise, it could lead to confusion, and if many bases are projecting at a given time into the active site, wrong bases might get incorporated. So, this finger domain ensures that a correct base is incorporated each time a reaction is taking place. The thumb domain also plays an important role in this particular process. Though it does not carry out any catalytic reaction, the thumb domain has residues which interact with the phosphate backbone of the newly forming DNA, which helps in maintaining the correct position of the primer at the active site, so that the incoming nucleotide only come to the primer site and make interaction with the primer. Also, the thumb domain makes strong association between the DNA polymerase and its substrate, so that once DNA polymerase is bound to the primer template junction, it carries on addition of a great number of nucleotides in one go. This is termed as processivity. Processivity is enhanced not only because of this particular aspect of thumb domain, but many other proteins which are involved in DNA replication also help in enhancement of processivity. The processivity is basically in this particular reaction is getting enhanced because of the interaction between the phosphate backbone of the newly formed DNA and the thumb domain, and also the interaction between the minor groove of the DNA and the palm domain. Now, what happens if a wrong base gets incorporated in the primer against the template? Because many times there might be a tautomeric shift in the bases, which can lead to incorporation of a wrong nucleotide at any given position. If it is continued, then it might lead to mutation in the gene and wrong proteins will be formed. However, DNA polymerase has another character, that is the exonuclease activity. DNA polymerase can bring about the excision of wrongly placed base from the 3' end. That is, it has 3' exonuclease activity residing in the same polypeptide itself. But this particular activity is far away from the active site of the DNA polymerase enzyme. Whenever there is a mismatch nucleotide present, then the rate of catalysis decreases to a very great extent, because correct interaction between the minor groove of the DNA and the palm domain cannot be formed. Whenever this happens, there is the movement translocation of the incorrectly placed nucleotides into the exonuclease domain. Herein, a series of nucleotides are excised by the exonuclease activity, and then the correctly formed primer template junction is translocated back to the active site for further reaction to take place. This is how DNA polymerase ensures that no wrong nucleotide is incorporated against any of the base of the parent strand, and this leads to a great decrease in the rate of mutation. Now, I come to the second type of enzyme, which is helicase, and it is also very important enzyme, because this enzyme is the one which will unwind the double stranded DNA for a primer template junction to form, and subsequent reaction will be carried on after the single stranded regions of DNA have been exposed. Now, the helicases were the first helicase was discovered in E. coli in 1976 and in lily plant in 1978. This particular set of enzyme belong to AAA plus family of protein, which is basically a family which can utilize ATP for carrying out a variety of functions. Helicases can be monomeric, dimeric or multimeric, and another characteristic of helicases is that they are processive. That is, once bound to DNA, they will bring about unbinding of the double stranded DNA, of a major stretch of double stranded DNA. Now, I will be talking basically about the prokaryotic helicase and eukaryotic helicase. The prokaryotic helicase has 6 subunits, and each of the subunit has ability to bind ATP. Now, how does this particular protein bring about unbinding of DNA? It is because it can slip on the DNA molecule. As can be seen in the figure, there are 6 different labels, namely 1, 2, 3, 4, 5 and 6. These are 6 different subunits interacting with the single strand DNA at 6 different positions. The helicase have DNA binding loops as well as ATP binding loop. Now, as can be seen, the 6th subunit is foremost foremost toward the 5' direction. However, as soon as the last subunit, that is the 1 subunit, binds ATP, there is change in conformation and it shifts toward the foremost position toward the 5' site. Later on, when the second subunit binds ATP, that will also shift its position toward the foremost position in the 5' site. Effectively, there is hand by hand movement of the different subunits on the strand of DNA, which brings about unwinding of the DNA molecule. These helicase molecules have definite polarity, that is, once they are loaded onto the strands of DNA, they can either move from 5' direction to 3' direction, or they can move from 3' direction to 5' direction. As seen in the given figure, almost all the prokaryotic helicases move in 5' to 3' direction, whereas the eukaryotic and archibacterial helicases move from 3' to 5' direction. The prokaryotic helicase is made up of six subunits. It is a product of DNA B gene, which codes DNA B protein, and 6 such proteins assemble together to form a hexameric ring shaped structure which is having threefold symmetry. One subunit is of about 471 amino acid residues in prokaryotic helicase, and it is also termed as DNA B. The loading of prokaryotic helicase onto the DNA strands is done with the help of another protein known as DNA-C. DNA-C is a helicase loader protein. It is again an AA plus protein which can bind ATP and carry out loading of the DNA. So, DNA-C binds to DNA-B and it causes loading of opening of the rings of the helicase, which is then loaded onto the single stranded regions of DNA. DNA-G, which is a primase in prokaryotes, it then binds to this particular complex and leads to formation of a primer. After binding of DNA-G, the ATP which are bound to DNA-B are hydrolyzed and DNA-B releases its grip on DNA. DNA-C releases its grips on DNA-B and it falls off this particular complex. And DNA-B is now free to carry on its action of unwinding the double stranded region of DNA by utilizing ATP. The eukaryotic helicases are also hexameric structures and they are named as MCM27 complex, which is basically a short form for mini chromosomal maintenance protein 27 complex. There are 6 proteins involved in eukaryotic helicases and all of these are coded by 6 different proteins. However, they have a similarity regions which is mostly in the region which is the nucleotide triphosphate binding region. They also have a symmetry, a 6-fold symmetry, and they also have a groove wherein the single strand of DNA will fit during the unwinding of DNA. The eukaryotic helicases require certain proteins known as CDC6 and CDT1 for loading onto the DNA strand. However, instead of a single hexameric molecule, a double hexamer is loaded onto double stranded region of the DNA. A complex of protein known as CMG protein complex is required for loading of the single hexameric proteins onto single stranded DNA region. This CMG protein can be bound to ATP or it can be bound to ADP. When it is bound to ATP, it is not able to load the MCM27 complex onto the strands of DNA. However, when ATP is hydrolyzed, then it is bound to ADP and it leads to change in the conformation of the helicase protein and also the strands of DNA, so that a single hexamer gets loaded onto each of the strand of the DNA molecule. So, all in all, helicase bring about unwinding of the DNA double stranded molecule using ATP, energy of ATP, and this brings about separation of the 2 strands, which is an essential requirement for formation of a primer and assembly of the replisome complex later on. After the 2 strands of DNA have got separated, to retain them in this particular conformation, certain proteins are required. these proteins are known as single strand binding proteins, because if we leave the two strands as such, they will again start forming complementary base pairing. Also, if the two strands are left naked, they will be subjected to digestion by different nucleases. In order to protect these strands from digestion by nucleases and for making them available for replication to occur, single strand binding proteins coat these particular strands of DNA which are single stranded at this particular juncture. The binding of single stranded proteins is cooperative in action. That means if one SSB protein has bound to a given strand of DNA, then it will facilitate binding of other proteins to that particular strand, eventually leading to coating of the entire strand. The single strand binding proteins help in keeping the DNA strands in extended conformation, so that the bases are exposed whenever there is formation of complementary base pairing with the incoming nucleotides. In E. coli, these proteins are known as SSB, whereas in prokaryotes these are termed as replication factor A, and there is a large family of these proteins. Once the two strands have separated and they have been coated by SSB proteins, the next step is formation of a small primer to which incoming nucleotides will be attached. Now, though DNA polymerase are like very versatile enzymes, they do not have the ability to start the synthesis of DNA strands by themselves. They always need a presence of a primer for this synthesis to occur. And because the DNA polymerase are not able to do so, another set of enzymes known as primases are required for formation of primer. Primases are basically RNA polymerases which carry out synthesis of a RNA primer using the information present in the template strand. These RNA primases are present in both prokaryotes and eukaryotes, and they lead to formation of about a primer of about 4 to 15 nucleotides in length. After the formation of primers, DNA polymerase takes over, and this particular primer is extended by the activity of the DNA polymerase by addition of deoxyribonucleotide phosphates. However, this particular protein is active only in the presence of certain other proteins like helicase when they form a primosome complex, so that there is no random activity of primase in the DNA. Only when a helicase is present, only when replication is going to occur, this primase enzyme will lead to formation of a primer and replication will initiate. However, because of formation of RNA primer, there occurs a peculiar problem that the DNA molecule which will be formed later on will be having ribonucleotides in it. For overcoming this problem, there are certain enzymes which excise the ribonucleotides from the 5' end. One such enzyme is RNase H, which leads to excision of ribonucleotides from the 5' end. However, it is not able to cleave the bond between the last ribonucleotide and the deoxyribonucleotide. For this, another enzyme known as exonuclease is required, which can cleave this particular bond. After the cleaving of the RNA primer, again the structure resembles a primer template junction because there is a double stranded region and a single stranded region. This primer template junction will now be utilized by the DNA polymerase to carry on addition of nucleotides, leading to formation of a double stranded DNA. However, another problem surfaces here, that the last nucleotide which is added is having free OH group, but the nucleotide which is present in the pre-existing strand of the DNA, it does not have 3 phosphate groups, it has only 1 phosphate group. So, how does this reaction take place? That is the ligation of the newly formed strand with the existing strand. This is brought about by another enzyme known as DNA ligase. Now, DNA ligase utilizes ATP and it transfers AMP from the ATP to the pre-existing nucleotide which has only 1 phosphate group. This in effect leads to formation of 2 phosphate groups along with ribose and adenine at the site where previously only 1 phosphate group was present. Now, the OH group of the newly formed strand will attack the phosphoryl group, leading to formation of a phosphodiester bond between the newly formed strand and the existing strand, and AMP will be released as a result of this particular process. So this is the mechanism of action of the ligase. It helps in the joining of the nick between the two nucleotides with the help of AMP molecule. Thus, there are no more nicks present in the DNA molecule. Everything is sealed, and that molecule is a perfect double-stranded structure. Whenever replication occurs and helicase is doing the unwinding, during unwinding, the region which has not been unwound till now, it gets supercoiled because of the torsional force. Now, if this supercoil were to exist for a long duration, then effectively all the unwinding activity will stop and DNA replication will stop at one point or another point. This is a problem faced in circular DNA of both the eukaryotes as well as the linear DNA of circular DNA of prokaryotes as well as linear DNA of eukaryotes. This problem is overcome by use of certain enzymes known as topoisomerases. Topoisomerases bring about the unwinding of the supercoiled DNA. Either they cut a single strand of DNA or they cut both the strand of DNA, and they swivel the region which has been supercoiled. This swiveling action leads to unwinding of the supercoil, so that this particular DNA is no more coiled extensively and is available for replication. Now I come to the last and one of the most important proteins in the series of proteins I have been discussing about, and that is sliding clamp protein. The sliding clamp protein is essential for enhancing the processivity of the DNA polymerase. It is basically a hexameric structure. In prokaryotes, it is formed by a dimer of three proteins, whereas in eukaryotes, it is formed by a trimer of 2 proteins. In prokaryotes, it is known as sliding clamp protein beta, whereas in eukaryotes, it is known as pCNA protein. The sliding clamp protein is loaded onto the single strand of DNA by sliding clamp loaders, which are again AAA plus binding proteins. These proteins have the domains which can bind to ATP, and they can hydrolyze ATP to carry out specific action, which in this case is binding to the sliding clamp protein and opening 2 subunits so that they can be loaded onto the single stranded regions of DNA. Whenever sliding clamp loader are bound to ADP, they are not able to bind to sliding clamp. However, once bound to ATP, they will be able to bind to sliding clamp and open the sliding clamp subunits and load them onto DNA molecule. This is how the mechanism takes place. Here a sliding clamp loader which is bound to ADP, it is not able to bind to sliding clamp, whereas when it is bound to ATP, apart from binding to the sliding clamp, it also causes its opening and its loading onto the DNA. So the overall mechanism helps in loading of the sliding clamp onto the DNA. The sliding clamp helps in increasing the processivity of DNA polymerase by keeping DNA polymerase in its position. If sliding clamp was not there, then the processivity of DNA polymerase would decrease many folds. Because it holds the polymerase in position, it leads to enhancement of processivity so that DNA polymerase adds a number of nucleotides at one go. When the DNA polymerase reaches the double-stranded region, it falls off from the DNA. However, sliding clamp stays there because many other proteins which are going to modify the DNA are also recognizing sliding clamp. They recognize the same domain of sliding clamp which is required for binding of the sliding clamp to the DNA. When these proteins have carried out their action and there are no more proteins to bind to sliding clamp, then sliding clamp also opens up and it falls off the DNA polymerase. Thus, all these enzymes ensure that DNA replication takes place effectively and in a very high fidelity manner. Now, talking about the direction of the extension of the newly formed DNA strands, as we already know that the DNA is a double stranded structure and the 2 strands are anti-parallel in nature. the newly formed DNA strand is always extending from the 5' to the 3' direction, because the 3' OH group can only attack the incoming DNTP. So, both the strands, which are the leading strand and the lagging strand, will be extended from 5' to 3' direction. Wherever the replication fork is moving in a particular direction, the leading strand will be synthesized continuously, whereas the opposing strand, which is getting exposed in stretches, will have Okazaki fragments synthesized complementary to it. Again, same set of enzymes will be used, but here RNA primase will be used a number of times more than that being used in the leading strand. In leading strand, RNA primase is used only once, because only one primer is required, but in the opposing strand, because the DNA is getting exposed later on, a number of RNA primers are going to be made, and RNA primase will be required number of times. So, the lagging strand will be formed in fragments known as Okazaki fragments, which will be later on joined together by DNA polymerase, and the two strands will be synthesized. So, today this brings to the end of the lecture today. Tomorrow we will be talking more about the models of DNA replication.
With this note, thank you, sir. Thank you so very much for giving us deep insight into the very topic, that is DNA replication. As we have some few minutes left with us, if we talk about DNA replication, and if you want to give a brief definition of DNA replication, that what replication is, then how would be defined?
It is basically copying of a DNA molecule into two daughter molecules. In these two daughter molecules, there are parent strand as well as the newly formed daughter strand. So, it is basically formed because of semi-conservative replication. Two daughter molecules are formed, having one parental strand and having one daughter strand.
This was for the today's lecture. What else are we going to study in the forthcoming lectures?
Tomorrow, I will be talking about the details of the replication mechanism, how it gets initiated, and different models of replication mechanism in viruses, prokaryotes, etc.
With this note, thank you, sir. Thank you once again for delivering such a nice lecture. I hope that all the students who might have watched this lecture would be benefited from this particular lecture. Dear friends, if you want to give your feedback regarding this particular lecture, or if you have any queries, if you have any questions which you liked to be solved out after seeing this lecture, then dear friends, I would like to say that you can mail us at info.cec@nic.in. We would love to solve your queries the next time when Dr. Varunendra Singh Rawat visits our studio, that is tomorrow, as well as we are going to upload this lecture very soon on the YouTube, so that it becomes convenient for you to see this lecture the number of times you wanted, and afterwards also you can mail us your queries, you can send your feedback to us at info.cec@nic.in. So, dear friends, we would be meeting again tomorrow, and we would be conducting another lecture, that is the 4th lecture on molecular biology with Dr. Varunendra Singh Rawat. Till then, we say goodbye, take care. Thank you so very much. Thank you, sir.
Thanks a lot.